All right, moving along, woo woo, to immune responses. You guys are doing great. So, we've already talked about this a little bit, but let's go in even more depth about how the body responds immunologically to pathogens. Now we finally get to talk about complement. But the first slide has to do with acute phase reactants. So, all the proteins that are released when there is acute inflammation. What happens? Just very briefly, so the liver is responsible for making almost all of the proteins in your body. So that is where these acute phase reactants are released from. And what are they? What are we talking about here? Like, there's a bunch of inflammatory proteins.
C-reactive protein.
Fibrinogen, like clotting proteins, iron proteins like ferritin and hepcidin. All these proteins are released whenever there is inflammation. And what is the interleukin that's responsible for the release of these from the liver? Interleukin 6. Very good. We'll talk about interleukins in a little bit. All right, finally, time for complement. So again, remember, complement. It's a system of proteins that complement, that's why it gets its name, complements the innate and adaptive immune system. Okay? There are three main pathways, the classical, alternative, and lectin. And they're very similar, so we'll just really talk about one to start off with, and then you'll get the hang of it. Start off with the classical complement pathway. Here we have a blood vessel, all right? Sample blood vessel. Here's a bacterium. Whenever there's an antibody, specifically IgG or IgM, that's how you can remember it. GM makes classic cars. IgG and IgM are associated with the classical complement pathway. All right? Got that? So, whenever there is an IgG or IgM binding to an antigen, such as this bacterium here, guess what? We activate complement. The classical complement cascade. Ooh. All right. So, what is complement? Well, like you just started to see, it's a system of proteins. The first one, which is C1. C1 binds first, then C2, C4. Don't really worry about all the numbers, but just know that there's a few C's that start to bind, 1, 2, and 4, and then they turn into this complex. This complex that, another name for this, do you know what it is? C3 convertase. This is actually kind of important. So C3, this is where things start to heat up. C3 gets converted into C3A and C3B. These are things that you actually need to remember. C3A is important because the A stands for anaphylaxis. You can think of that. Because anaphylaxis means like dilating blood vessels. Well, that's what it does. It vasodilates. That's what C3A does. And C3B, what's special about that, what do you think? It's actually an opsonin. So just like IgG is an opsonin, remember what that is? Opsonization is allowing and facilitating macrophage consumption of pathogens. Remember, it's like sticking a toothpick into a slippery bar of soap. It allows macrophages to grab on to that toothpick and consume that slippery bar of soap. I don't know why they'd want to do that, but that's how you can think of it. So C3b and IgG are opsonins. Also, C3b, another random correlation, is that it assists in the removal of immune complexes. So the removal of antigens and antibodies. All right, so continuing on here. So then comes C5. Here's another important one. So things to remember are C3 and C5 so far. Well, another name for this complex that we see here is C5 convertase, because it's going to convert C5 into... C5 gets turned into C5a and C5b. And C5a itself is, think of the A for anaphylaxis, because it also contributes to vasodilation, just like C3a did too. The A's are for anaphylaxis. But it does something else that's special as well. Do you know what that is? It actually calls in a type of cell, and that cell is a neutrophil. So it's chemotactic to neutrophils. Neutrophils come in, and they, remember what they release? They release those reactive oxygen species that tear up everything in sight. So, good things, bad things, they don't care. They just want to destroy things. All right, so Then after C5b, come some more proteins. C6 and C7, C8 and C9. These 5 proteins here represent MAC, okay, the membrane attack complex. You see how it's in the shape of a pore here? It's because it drills a little hole in the membrane of wherever it's binding. So in this case, the bacterium. There's a little pore that's formed. It causes this bacterium to lyse, things to rush out, and you have destruction of that. So pretty cool, right? So complement, very important, very, 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 very important to know for your tests. Next up, the alternative pathway. So we just talked about classical complement. The alternative complement pathway is essentially the same thing. It's just a different thing that's triggering complement. So we talked about classical where it was an antibody. This time it's just a bacterium. And specifically, inside the bacterial cell wall, there is a fragment called lipid A, or endotoxin. Endo means within, toxin. So it's embedded within, and it triggers the alternative complement pathway. All right, so again, it's just going to be a review. It basically is the same thing, except slightly different products that begin the complement pathway. So this time it's called C3H2O. Then there's a B and then a D. The way I remember this is the B is different. I mean, these are too, but I remember the B because B for bacteria. So these all turn into this C3BBB. So all these Bs make me think of bacteria. That's how we differentiate it. And another name for this is just like that other product in the other pathway. This is a C3 converting protein. So it's going to convert, see that bacteria equals BB. So it's going to also convert C3 into C3A, C3B, just like we saw before. Same exact things from here on out. C3A is an anaphylotoxin. That's what the A stands for. Vasodilation. This is a C5 converter now. And we're going to have C5A, which is also an anaphylotoxin. And it's also going to do what, again? Call neutrophils, release the reactive oxygen species, and tear up the tissue. We have C6 and C7, C8 and C9. This is called what, again? It's a nice little review. This is called MAC, membrane attack complex. Pokes a hole in the bacterium. Boom. Lysis. Pretty easy. You got it now, right? Good. All right, now let's talk about what can go wrong with the complement cascade. And in general, it's a problem with inhibiting it, so it's always activated. Why is this a problem? Well, look at this poor kid. Look how swollen he is. This is called angioedema. Why is he so swollen? Well, when we don't inhibit complement, we have constant activation of it. And remember those anaphylotoxins like C3A and C5A? Yeah, those are always activated, and those lead to angioedema and swelling and vasodilation like we see here in this poor kid. So what's the reason for this kid's deficiency? Well, it's a genetic problem, and he can't break down C1. So
the C1 esterase that normally breaks it down is decreased.
And you have constant activation of C1 and constant C3A, C5A anaphylaxis. A similar thing can happen with C3. C3, remember, it's broken down as C3b, C3a. Well, certain people can actually have a deficiency in C3, and this time they actually don't have enough complement being activated. So they actually are susceptible to infections because they don't have enough inflammation around. And what else was special about C3b? It's an opsonin, right? So they're going to have decreased opsonization. That'll lead to infections. And C3b is also helpful in what? Clearing immune complexes. So immune complexes are an antibody plus an antigen. And so if you can't clear these, you're going to have increased immune complexes. What type of hypersensitivity is this? We'll talk about that later. That is type 3. All right. So that's the problem with C3. All right, another thing is you could have a deficiency in the membrane attack complex. So remember, that is C5b, C6, C7, C8, and C9. Remember this whole thing? So that normally punched a hole in pathogens. Well, you don't have that around, you run the risk of susceptibility to a certain type of bacteria in particular, and that is, it's encapsulated, it is Neisseria meningitidis. People who are deficient in this membrane attack complex are particularly susceptible to Neisseria meningitidis dissemination all over their bodies. All right. And finally, here we have a red blood cell here. There is something that normally breaks down complement. Otherwise, you would have it going on all the time, just like with that C1 esterase, right? So this is called the decay accelerating factor. It's on the surface of almost every type of cell. It prevents complement from attacking your own cells. So some people are lacking this decay accelerating factor, and thus complement can attack your own cells, and that is a problem. That disease is called paroxysmal nocturnal hemoglobinuria, because we just saw there that red blood cell was killed, and the hemoglobin will leak out into the bloodstream, and you'll urinate it out. So, especially at night. That's why it's called paroxysmal nocturnal hemoglobinuria. Flash quiz time. What are the complement proteins that trigger anaphylaxis? Definitely know this, but it's just very important. That's why I'm asking you. So the A proteins, C3A, C5A. What else does C5A do? C5A calls in neutrophils. Good.
All right, so now let's talk about how cells communicate with one another. And they do this through cytokines, or cell connections. And a specific type of cytokines are actually interleukins. What does this word mean? Inter means between, and leuc means white, so between white cells. This is how specifically white cells or immune cells talk to one another. There's about 12 of them that you should know. The first 5 can be remembered using this helpful mnemonic, hot T-bone steak. Sounds kind of silly, but it's actually a very helpful way to remember them. So the first one, interleukin 1, you can think of hot for fever because it makes you hot. How does it do this exactly? Well, where's your thermoregulatory center? In your hypothalamus, right? So it actually releases prostaglandin E2 in your hypothalamus and makes you hot. All right, interleukin 2, you can think of the T because it stimulates T cells, as we talked about before. Interleukin 3, bone marrow stimulation, all the cells in the bone marrow. Okay, red cells, white cells, everything. That's what interleukin 3 does. Stimulates your hematopoietic cells. Interleukin 4 and 5, you can think of the letters E and A of the word steak because they stimulate the class switching of B cells to instead make IgM, they make IgE and IgA, respectively. So what else does interleukin 5 do? There's something special about this. It's chemotactic to a certain type of cell, and that is eosinophils. So interleukin 8, you can remember thinking of cleanup on aisle 8. Okay, cleanup on aisle 8, that is because, what is it? Chemotactic to neutrophils, right? There's something else that we talked about was chemotactic to neutrophils. What was that? Part of the complement cascade? C5A. Yep, very good. So continuing on with the cytokines, interleukin 10, what does that do? We talked about that earlier. It's an inhibitory cytokine released by, remember what type of cell? T regulatory cells, right? So, and also by Th2 cells to stimulate Th2 but to inhibit Th1. Okay, so that's what interleukin 10 does. Interleukin 12 is the opposite. Remember 1 before 2 because it stimulates Th type 1 cells and inhibits Th type 2 cells. Okay, so you can remember that. And macrophages as well. So we'll talk about that in a different section, but it does actually stimulate macrophages. Interferon gamma, who made that? Remember the gamma is kind of like a bent T, so that's made by Th1 cells to stimulate macrophages. Remember this co-stimulation? What did macrophages make to stimulate T cells? Tumor necrosis factor alpha. That's what they made. So these cytokines, as we remember, stimulate one another. All right, let's keep going with cytokines and now talk about some more interferons. So we mentioned interferon gamma already, but there's also an alpha and a beta. What do these do? So they are essentially very antiviral. They put your body in an antiviral state when we're talking about especially alpha and beta interferons. So how do they do this? Well, they stimulate the synthesis of ribonucleases, all right? And what these enzymes do, remember enzymes always ended in ACE, what this enzyme actually does is it tears up viral genes. The DNA and RNA just degrades it. Okay, so that's very helpful. Interferon gamma, we talked about that already. Remember, it was made by what cells? T cells, primarily, and it stimulated macrophages. But what else does it do? It actually has anti-tumor properties. So here's MHC1 receptor. And remember, MHC1 is the receptor that does what? It presents fragments of tumors or viruses to immune cells so that it can be destroyed. By what types of immune cells specifically? Natural killer cells and cytotoxic T cells. Very good. So what interferon gamma does is it actually increases the synthesis of these MHC receptors, so you have a much greater chance of presenting the enemy to these immune cells and then thus being protected from them.